So, Bryant is here, Curry is online, Darby is here, Fisher is not here, Hamilton is online, Melvin is not here, Ashley is here in red, both of the Millers are online, Norris is here, Rollins is not here, Saunders is online, Chamblin is here in red, Vargas is not here. See anybody? All right, let's be good. Pick back up where we left off. I want to remind you. I meant to remind you, or make the announcement Wednesday. We are skipping the chapter after this, so we are not doing chapter 10. I forgot to mention that. Chapter 10 is one of those new chapters that was not included in the textbook, and I was only going to include it in this semester if we were ahead of schedule. We're not ahead of schedule, pretty much right where we should be. Therefore, again, we're going to skip chapter 2. You could read it. It's still really interesting stuff. It's actually really relevant to your everyday life because it has things informationally to understand GMOs and like the COVID-19 vaccine and things like that. So that's the one thing I wanted to let you know. We are skipping chapter 10. This is the last chapter for this exam. And what else is there? Oh yes, as I said, I've been a written announcement. I didn't verbally say it. But at the end of this chapter, we're going to talk about, or at the end of this chapter, at the end of this presentation, we discuss viruses briefly. And in your book, that doesn't come until chapter 17. So when you're studying that portion of this PowerPoint, you're going to go to chapter 17. So any questions? Okay, let's get back into it. This is where we left off last time. First word for attendance is going to be the one I circled right here. So do it like I did on Wednesday. If you're watching the video, so not if you're live streaming or if you're here in person, but if you're watching the video, send me, instead of sending me that word, send me a sentence that uses that word. Anyway, like I said, this is where we left off. We were talking about DNA polymerase because we're talking about DNA replication. The DNA polymerase, that's the enzyme that does it. Some things that we don't get into that your book gets into that I said, first of all, like I said on Wednesday, the DNA itself has to get opened up, right? Because the code is right there where the nitrogen space is, right? Those are the codes. So we have to open it up to get to the code. Now, your book talks about the enzyme that does that, and I don't require you to know that. The book also mentions that when the DNA opens up, I'll try to draw it. This is going to look like garbage. You have double strand, and it opens up right there. There's your nitrogenous bases hanging off, and those nitrogenous bases, that's the code. But your book mentions that the DNA polymerase can only work in one direction. Remember, there was this 5' stuff, 3' stuff that I said you didn't have to know. I stand by that. I'm not going to ask you that. But basically, let's just say DNA polymerase can only work in this direction. And that means the other one can only work in this direction. Your book explains, there's a whole paragraph on that. It explains, generally it works pretty simply. It just runs down the DNA, it reads the code, and it brings in the new nucleotide. But your book does mention that there are some exceptions to that because the other one is a little bit more complicated. The only reason I'm bringing it up is for you to take notes and write a note to yourself and say, I do not need to know anything about that. Okay, so in long and short, anything I don't tell you on this portion of the lecture, you do not need to know. You can read it in the textbook, I'm not going to text you. Anyway, back to the DNA polymerase. All this stuff we already said, this is the slide we left off on Monday or on Wednesday. But to put it visually, again, like I said, there's an enzyme, helicase, just so you know, you don't have to know it. It unwinds the DNA, it opens it up, you can't see it in this picture. DNA polymerase attaches, you can't see it in this picture either. It's reading this, right? So it reads that there's an A, so it's going to bring in a T, because T is complementary to A. Then it reads that there's a C, so it's going to bring in a G, because G's are complementary to C. And that's what we're left off. And again, like I said, a good test question is, when this happens, once we duplicate one strand of DNA or one double helix of DNA, and we have two of them, what do they consist of? And the answer is each one consists of an old strand and a new strand. You need to know that. There's one test question on that. And on this slide, there's not much you need to know for the exam. And again, your book goes into greater detail, but I'm not going to test you on it. So you already know this part. DNA replication happens in the S-phase of interphase. Yes, you need to know that because you taught that in the mitosis meiosis chapter, and here it is again, right? So 2 chapters on this exam, you've been told this, so you need to know this. DNA replication happens during the S-phase of interphase. And then I might even ask you this, where on chromosome does it happen? Where does it start? It starts at places called the origins of replication. I will point out, as your book points out, what we're talking about right now is eukaryotic chromosomes, right? So we're talking about more complex things like humans or fungi or plants. If we were talking about prokaryotes, which your book does, and we don't. If we talk about bacteria, it's a lot simpler, so they don't have all this stuff. But anyway, it starts at these places called the origins of replication. So there's a code there. Your book even talks about what the particular code is, but you don't need to know it. But anyway, the DNA polymerase can read the code, right? The A's, the T's, the C's, and the G's. There's a certain code that tells that enzyme, okay, this is where I need to attach. So it attaches at the origins of replication, moves, doing what I showed you before. It's reading the bases, it's bringing in the new complementary bases, it's building a new strand of DNA. And that creates something called replication bonus. But I'm not going to ask you anything about replication bonus. And again, just so you know, I'm going to ask you, these happen at the origins of replication. So let me draw a chromosome here. Try to. That's the chromosome. It's not like the DNA polymerase attaches to one end and makes its way all the way down to the other. That's not what happens. What happens instead is, draw the chromosome again. There's different spots it attaches, right? Maybe one will attach here, maybe one will attach here, maybe one will attach here. Basically, we have multiple DNA polymerases doing the job. Not that I'm going to test you on that. I just wanted you to know that. And your book also points out that I'm not going to test you on this either. There's certain things that kind of regulate. These origins of replication, they're not all the same. Some of them have these genes constantly going. Well, no, I'm sorry. We'll talk about that later. That's the next section. I'm jumping ahead. Anyway, any questions about this slide? Okay, if you download the PowerPoints, you can see little animations of this. I recommend watching them. So this shows the origins of one origin of replication, one of many, and it shows how that works. Then it gets into things, oops. Then it goes into some terms that I'm not covering. Again, your book discusses it, but I don't. So you can read about lagging strand, you can read about leading strand, but I'm not going to ask you anything about that. And also, I should point out that picture I just drew most recently was actually incorrect because I drew it the wrong direction. So DNA polymerase always goes from 5' to 3'. Your book discusses that, but I'm not going to test you on that because I remember being a freshman taking this class, the biology major version of this class. Even that confused me. It took me a while to get 5', 3'. So instead of focusing on the big picture, I was just stuck on the whole 5', 3' thing and didn't really learn what I should have learned. So forget the 5' thing, unless you understand it, but I'm not going to test you on that. Anyway, that's it for the video. So any questions? That's it for DNA replication. Are there any questions about DNA replication? Okay. Now for the more relevant, depends on how you look at it. I think this is more relevant. There's a lot of technology out there, again, chapter 10 that we're skipping. There's a lot of technology that really focuses on what you're about to learn. Going from DNA to RNA to protein. So any kind of GMOs, right? This is what we work with, right? Because we want to put a gene into corn that makes it resistant to certain insects. Well, you're going to need to, again, put in a gene so that you put it into the DNA, so then turn it into RNA, or turn it into protein, so on and so forth. So here we go. As you should already know, this isn't necessarily going to be a test question, this is more of an introduction to what we're about to talk about. DNA provides instructions to a cell, obviously, right? So again, going back to my analogy that I've been using for a few days now, you think of your chromosomes as books. right? In the case of humans, we have 23 pairs of books, right? And what's in those books? Instructions. Instructions on how to build proteins. And those proteins, as we know from earlier chapters, do all kinds of things. In this case, sometimes they provide instructions for a cell. Sometimes they actually provide building material for a cell too. And obviously, if there's a multicellular organism, DNA can also provide instructions to the entire organism. You already know this. We've already talked about it. We've just never talked about it in this context. So that last lab we had with genetics, we talked about, I keep using the most simple one that I can think of, is the chin dimple, right? You either have it or you don't. That's because there was one gene where you either had this allele or you had that allele, and then from there, that provided instructions to your whole body on how to build a chin, right? So again, the gene makes up your genotype, then that, of course, as you learned from the last chapter, results in your phenotype. So how does this happen, right? Because what's your chin made of? Does anybody know? Forget the skin and all that, but what's your chin made of? Here's an anatomy question for you. Don't overthink it. I'm not getting complicated. Like really, I would ask a fifth grader, what's your chin made of? And they'd get it. They wouldn't overthink it. Yeah, bone, right? Bone's not protein. So then the question is, like, how does that work then? If genes provide instructions on how to build proteins, how is it giving us a chin dimple or not a chin dimple? And that's what we'll talk about briefly. I'm not going to focus on it too much, so there definitely won't be any questions on it. But I just want you to understand it. It's important to understand. Because again, here you learned all these things about how genes affect phenotypes, right? Flower color, freckles, ear connection, interlocking thumbs. And those things aren't proteins, right? So hopefully if you're a thinker, you're like, well, how does this work? How do genes control all these things if they're not proteins? Well, again, as I'm saying, as you should know from the last chapter, what is a phenotype? Your phenotype is a physical trait, right? So again, smooth chin or not smooth chin, blue eyes or not blue eyes, bent little finger or not bent little finger, red flower or white flower, right? Those are your phenotypes, or those are the phenotypes. And those come from the actions of a wide variety of proteins. This is what I'm getting at here. So yes, a lot of proteins are actually used, right? So your fingernails, that's made of protein, right? The muscles got a lot of protein in it. So yes, sometimes the gene does code for a protein that's just directly used as it is. But more often than not, it's this right here. You should be familiar with these in a few of our chapters, right? Remember, enzymes are proteins. So we have genes that code for these proteins that make up the enzymes. In a sense, again, going back to my analogy, we have these books of instructions on how to build proteins. You can think of it as if this was a construction thing or a factory thing. You have a book of instructions on how to build different products, like how to build, I don't know, fidget spinners, because I used that analogy earlier in the semester. Instead of opening the book, and it teaches you how to build a fidget spinner. If you open the book, what it actually gives you the instructions on how to build the machine that builds the fidget spinners. Same thing here, right? So for your chin dimple, the gene didn't directly make your chin dimple or your smooth chin. What your gene did was build a protein. That protein was an enzyme. That enzyme catalyzed these chemical reactions, and it controlled whether you had a chin dimple or not. Now, granted, I am oversimplifying a little bit, but for the most part, that's how it works. So again, all these things that you've learned, we've already learned about a lot of enzymes, right? In photosynthesis, there was a bunch of enzymes involved. In respiration, there was a bunch of enzymes involved. DNA polymerase is an enzyme we learned in this chapter. RNA polymerase is an enzyme we're about to learn in this chapter. All these enzymes, again, they're made up of proteins. So there are genes that control this, right? Genes that have the instructions on how to build these enzymes. Without those enzymes, you don't have life. Speaking of which, that'll be the next 10th word. I'm circling it, pointing at it. Please send that in a sentence if you're watching the video. So not if you're here, not if you're watching the live stream, but if you're watching this video some other time. Can someone online confirm that you can hear me? Sorry, I should have done this earlier.
We can hear you.
Thank you. Wanted to be sure. I don't want to go through that again. Anyway, any questions about this slide? All right, the next slide is an overview. So you don't necessarily need to write anything down yet. Again, it's an overview. This is the big picture. So it's an introduction. As I keep saying, DNA, right, your genes, it specifies protein synthesis. The new information I'm giving you that you need to know is it happens in 2 stages, transcription and translation. You need to know that. That's the first thing I think you should memorize. Again, everyone learns differently. My recommendation when you're studying this is a lot like how I recommended studying photosynthesis, respiration, and mitosis and meiosis. Learn the big picture and then start filling in the details. So the big picture, the first thing is this bullet point right here. DNA synthesizes proteins, 2 stages. Remember that first, right? When DNA makes proteins, it happens in 2 stages. Then once you have that memorized, then memorize the names of those 2 stages. Transcription, translation. Then once you have that memorized, then you can memorize what that actually means. What does transcription? Oh wait, no, sorry. Then memorize the fact that it's always in that order. Transcription, then translation. And then once you have that memorized, then memorize what they do. So obviously, based on the fact that the chronological order of it, transcription is when we go from DNA to RNA. Translation is when we go from RNA to protein. And there will be a test question that says which of these is in the correct order. The correct answer will be DNA, arrow, RNA, arrow, protein, or polypeptide. The context of this conversation is the same thing. Remember, a polypeptide is just a long strand of amino acids, and then once it folds up into the right shape, then it's considered protein. Yeah, so these are the things you should memorize before you start worrying about the details. If someone's going to miss that question, it's going to make me really sad, because forget everything else. If you don't remember that the process goes from DNA to RNA to protein, then who cares what the names of the processes are, or all the details I'm about to give you from all these other processes. That's the most important. You need to know that we had the code in the DNA, that then gets transferred to the RNA code, which then gets turned into protein. Sort of like how people miss some of the photosynthesis and respiration questions that were really basic and made me sad, like chloroplasts are in plants, right? Or respiration happens in mitochondria. Those are the big things that people are more important than all those little details. And by the way, you can still retake that exam. Anyway, let's go back to this. So the relationship between genes and the proteins are one of information flow. That's not going to be a test question. Again, it's kind of an introduction to what we're about to talk about. And again, like I've already said, and you can see it again in writing, the function of a DNA gene is to dictate the production of a polypeptide. I will say this too. There are exceptions to this. Not that you need to know this. You can look this up for independent work. It's not going to confuse you. But as I keep saying, first we go from DNA to RNA to protein. Sometimes we just need the RNA. We don't even need the protein. So there are some genes that code from some RNAs that's like, all right, that's it. We've got the RNA that we need, and we're done. Right? So yes, generally speaking, it goes from DNA to RNA to protein. Sometimes we're just after the RNA, so sometimes it just goes DNA to RNA. Put that up for independent work if you want. Because you're about to learn, and this is a little something I should go ahead and warn you for this exam. There's going to be 3 different types of RNA that you need to know about. But if you look it up for independent work, there's actually a lot more different types of RNA, and they do all kinds of interesting things that we just don't learn about in this class. All right, so let's talk about how we go from nucleotides to amino acids in overview. So again, sort of like I did with photosynthesis and respiration, I'm going to give you the overview first, and then we'll jump in and look at the details. So again, The information in the DNA is transcribed to RNA, right? So that's that first thing you need to know, right? Step one is transcription. Because we're going from DNA to RNA. And then that information is translated to polypeptides. And then again, like I said, technically a polypeptide is not a protein, so it folds into its shape. But for the purposes of this chapter, I'm not going to differentiate between the two. Just because I want you to focus on the other stuff in the exam. So for this exam, proteins and polypeptides mean the same thing, just for the exam. We know that they're different, but I don't want you to have to stress about that for the exam. When you're studying, I don't want you to think, is he going to use proteins or polypeptides? And which one's correct in which situation? In this situation, in this exam, you can consider them the same thing. So again, that's just the overview. Here's a picture of the overview, not the details of the overview. This is not from your textbook, this is from your old textbook. Here you have your DNA, it's inside the nucleus. Like I said, the first portion is transcription, where we go from DNA to RNA. You can see this is all happening, that part's all happening inside the nucleus. And then that RNA, some things happen to it, we'll talk about what happens to it. And once it's ready, it leaves the nucleus, it goes out to the cell, out to the cytoplasm, where it's then translated into a protein. And I'm going to use the same analogy I've been using. Again, imagine genes are, books, right? Each chromosome is a book. You have a set of instructions, how to build stuff. Let's say in the library, we've got the master copy of how to build everything on campus, right? You just got one copy of each book. So one book, one copy, well, two copies, one for wrong. Yeah. But anyway, you've got the master copies, right? There are no other copies. And then you open it up and you go to whatever page to get instructions on how to build a bench. Like we need to build a bench on campus. So what you do is you take that master copy, since there's only one copy, you're not going to leave the library with it and go out onto the campus and build yourself a bench. First of all, that's too cumbersome. You don't need the whole book. You just need that one set of instructions that came from the book. Second of all, that's your only copy of the book, so you don't want to take that and risk taking it out of the library and have it getting, messed up. So what do? You make a copy of it. That's what transcription is. How many of you ever heard the word transcribe outside of biology? What's it mean to transcribe something? Does anybody know? Like if I was a boss and I had a secretary, and I told him, hey, I need you to transcribe this speech I'm about to give you. What am I telling my secretary to do? Write it down, right? So transcribe is to write, which makes sense. And everything I'm about to tell you, I'm going to tell you again, and I'm going to put it in writing. So this is, again, just an overview. Yes. So we need to make a copy of just that one portion of the book, right? So we're going to transcribe it. We're going to write it down. And you do it in the same language, right? So if you're or forget the analogy I just used. Let's imagine going back to the original analogy. You're the construction worker. You go to the library, you open up the book, the page, whatever, to get the instructions on how to build a bench. You open it up, it's written in English, you write it down in English, right? Like I said, first thing you need is this many 2x4s. You need this many saws, right? You write it in English, you're transcribing it. Then you take what you wrote, you leave the library, you go out onto Quad, and you're going to build it. And you're going to translate those written words that are in English. You're going to translate what's written there into a three-dimensional bench. You're going to follow the directions and say, all right, first I need this board, attach it to this bolt, then attach it to this bolt, and I put this board under here, right? So you're translating what was written down. Same thing happens here. That being said, now let's jump into it. Also, if you download the PowerPoint, there's another little video you can watch. This PowerPoint's chock full of little videos, and I think it's really cool to see it done like that. Anyway, like you keep saying, the first step is transcription. And then we talk about translation. So in a sense, we keep talking about languages, right? Like I said, with my analogy. The book's written in English, so you write it down in English, and then you take it out, and you actually use those words written in English to build some three-dimensional object. Well, let's go back to that idea, right? Let's use that analogy again, except now we're talking about the actual situation. So what is the language of nucleic acids? As you already know, but I'm saying it again and putting it in writing. In DNA, the language is the sequence of nucleotide bases. I'm just going to randomly make up some letters here. The DNA sequence was A, T, C, C, G, A, T, A, right? Excuse me, no need to write those down. I just randomly came up with some bases. So that's the language. So just like the English language has A through Z, right? The English language has letters. So does the DNA language. It's just a bunch of letters. Of course, again, we have A all the way through Z, right? But in this language, we only have A, T, C, and G. So this language only has 4 letters, and we already know that. What the book mentions, but I'm not necessarily going to ask you, is that a typical gene has thousands of nucleotides. You look that up for independent work if you want. What's the average length of a human? The average length of a human gene. Excuse me. Then you could look up, like, I don't know, what's the longest human gene? What's the shortest human gene? What's the longest gene of all organisms that we know of? What's the shortest gene of all organisms that we know of? If you want to do it. Anyway, this code, this language in DNA, these letters, are transcribed to RNA. Here's what you do need to know. I've said it before, I'll say it again. Take your notes and you haven't written this down yet, write it down. A's always go with T. C's always go with G. So for example, for the exam, I might give you this code down here, and I might say transcribe it. So you need to have these rules memorized. A goes with T, and C goes with G. You need to have that memorized, because then I'm going to ask you to transcribe this. So that's DNA right here. Of course, RNA doesn't have T's, right? So for RNA, it goes A's go with U, and C's go with G. You need to know that, because again, there will be at least one question where I cover the stuff we've already talked about, right? So DNA replication. So I might give you this code and say, hey, replicate this DNA. In which case you would just do complementary, again, A's with T's, C's and G's. There's going to be questions, at least one question where I say, I need you to transcribe this. So you take this code, and then you put in the complementary code to make it in RNA. And again, like I said on Wednesday, if you have any questions on the exam that ask you for RNA, or tell you to transcribe something, if it has a T in it, know it's the wrong answer. Because there are no T's in RNA, right? There's only U's. So when you're doing the exam, it'll save you some time. Look at all the options that have a T in it. Go ahead and cross those out. Those are not the options. Not going to be a correct answer. So really quick, let me do this. Let me transcribe this for you. So again, A's go with T's, but there are no T's in RNA, so there's a U here. T's go with A. C's go with G. C's go with G. G's go with C. Again, A's go with T, but there are no T's in the RNA, there's only U's. T's go with A. Again, A's go with U and U. That's what you'll have to do. And the good news is, again, these are multiple choice. I will say this too, because a handful of times since I've been doing this since 2016, a handful of times I have made a mistake in the multiple choices, and none of the answers are correct. So if you get to that question and you're like, none of these are right, I'm pretty sure none of these are right, just choose one and then move on, and then just send me an email or something. Say, hey, that question with the transcription or the DNA, there were no correct answers. And then I have to go in there and give everybody that. It happens occasionally. I guess the point is, if that does happen too, I'll catch it. I'll catch that mistake. So it's not like you need to tell me for that purpose. I guess the most important thing here is, again, if you see something written like that, and there are no correct answers, I just don't want you wasting time. Just move forward. Anyway, any questions about this so far? Again, this is still just the overview. So again, this is a language, just like the English language. And it uses letters, just like the English language uses letters. And in this case, this is how we're transcribing. Now, translation, that's a different story. That's the next step, right? Translation, just like it means in any other situation, it's going from one language to the next. In this case, we're going from the nucleic acid language to the polypeptide language. So that's why it's called translation. So hopefully that'll help you remember. Going from DNA to RNA, remember they're both nucleic acids, so it's the same language. So that way it's just transcription. You're not translating anything. You're just writing the same code down, from one place to the next. Translation is when you're not doing nucleic acid language anymore, right? You're building a polypeptide. You're translating it from one language to the next. As you should already know from previous, I think, exam 1, and again, it's coming back, you need to know it again. First of all, polypeptides are the polymers that make up the proteins, right? And the monomers that make up the polypeptides are the amino acids. And there are 20 of them. So again, this is a language thing, right? We're getting translated from one to the next. So it's the sequence of RNA bases that dictates the sequence of amino acids of the polypeptide. In other words, it's those letters, it's those codes in the RNA that tell us which polypeptide. So, and I'll show you a picture this will make a lot more sense later. Yeah, the letters of the RNA say, all right, bring me this amino acid. And then you read the next letters and it says, okay, now attach this amino acid. And then you read the next code and it says, okay, bring me this amino acid, right? You're just making a chain of amino acids. Because the letters in the RNA, those are words, basically, just like in the English language. They are words. Like, if you have the, well, I don't want to do that. I might confuse this. Like those three letters, DAD, right? DAD in the English language means dad, right? So you were trying to make a picture. You had a certain group of people in the picture in a certain order. And you might have the word dad. Like, all right, I need dad, I need you. And then the next word is MON. Like, all right, now I need the MON. Need you, Mom. Anyway, here's the picture of what I'm talking about. Here's a long strand of DNA. We've kind of zoomed into a small part of it. We're only looking at 3 genes. Now we're going to zoom into one small part of gene 3. So here's the codes, right? Again, there's the letters AAA, CCG. You don't, when you're studying this, don't memorize those letters. These are just random letters used as an example. So we need to transcribe. So we go from the DNA language to the RNA language. A's go with U, C's go with G, G's go with C, again, C's go with G, A's go with U. What I haven't taught you yet, and we're going to talk about this in more detail on the next page, but I'll go ahead and show you before we put it in writing. Like I keep saying, for this nucleic acid language, we deal with letters, just like we do in the English alphabet, the English language. But instead of having the whole alphabet, we only have those 4 letters, right? But just like in the English language, you can rearrange those letters to make different words. Of course, you're kind of limited to what you can do because there's only 4 letters. And yet, there's another rule that you have to know. Not only are we only dealing with 4 letters, but every word in this language is only 3 letters long. And it's called a codon. Right. So in the English language, you've got the word A on one hand, and then you've got like a word like utilitarianism. Right, so we have all kinds of different lengths of words in the English language. And in this language, every word is only 3 letters long. Actually, if you're good at math, you can do the math and figure out, hey, there's only 4 letters. There's only 3 letters in each word. How many words can we make? And the answer is 64. Not that you need to know that, but you can do that math and figure it out. But again, here we have these 3 letters. I just focus on these. These 3 letters of DNA say, all right, let me transcribe that to these 3 letters of RNA. And that is a word that tells something that I'm going to teach you about later. That tells something, all right, I need this particular amino acid. In this case, I need a square amino acid. And this word says I need a circular amino acid. And this word says I need a triangle amino acid. Again, this word, which is the same as this one, says bring me a square amino acid. But that's how it works. Of course, we're not with amino acids. We're not dealing with shapes like square, circle, triangle. They're actually different things, but that's basically how it works. You have these letters. These letters make up words, and each one of those 3-letter words dictates which amino acid is going to be added to the chain. Now that I said that, let's look at that in writing. Again, the flow of information from the gene to the protein is based on a triplet code. That triplet code, like I just showed you on that previous slide, they're called codons. Again, you have to think of it as the language. There's 4 letters in this language. All the words in this language are all composed of 3 letters. And again, as you already know, because we've already said this, and I just showed you the picture of it, the codons in the DNA are transcribed into complementary codons in the RNA. That's the same thing I've already said earlier, just a little bit more specific. Previously, I said the nucleotides in the DNA were transcribed to the nucleotides of the RNA. And this is still true, except now I'm being more specific and saying these 3-letter words, these 3-letter words, these 3-letter words. And then those codons in the RNA are then translated into amino acids, right? So this 3-letter RNA word says, bring me that amino acid. This 3-letter RNA says, bring me that other amino acid. That's how it works. The next word for attendance, I will put a box around it. The next word for attendance. Again, I'm just going to stick with the same theme. As usual, if you're here or if you're watching this live right now, just send me that word, either for extra credit for you guys or attendance points, participation points for those of you online. Or if you're watching the video later on, then send me a sentence with that word in it. All right, so any questions about this slide? And again, this is still just an overview. Sadly, it gets a little bit more complicated than this. There's the picture I already showed you. So, we've been talking about this code, we've been talking about this genetic language. Let's dive into it a little bit deeper here. Let's talk about this genetic code. Now, I'm not going to ask you the definition of the genetic code, but the definition is it's a set of rules that converts RNA sequence to the amino acid sequence. Now, I'm going to give you those rules. Yeah, that's what the genetic code is. Just like a language, right? Because again, we're translating something. If you've ever seen something like we had some kind of secret code, right? There was always some kind of way to decipher the secret code, like the Native Americans who helped in World War II, like the Marines who they spoke Navajo and other languages. The enemy didn't know that. Anyway, again, I've already told you this, but I'm not going to ask it. So, again, if you did the math, it's only 4 letters in this language. Each word can only consist of 3 letters. So statistically, or if you just went out and drew it, you can figure this out. That means there's only 64 words in this language. Of those 64 words, 61 of them say, hey, bring me this amino acid, bring me that amino acid, bring me this amino acid. And 3 of them just say, stop, we're done doing our job. Now, I will ask you about stop codons later, but one thing you don't need to know about stop codons is that there's 3 of them. So again, everything in this bullet point, I'm letting you know, I'm telling you, but I'm not going to ask you any questions about it. As you already know, I'm beating a dead horse here, a given RNA triplet always specifies a given amino acid. It's universal. And I'll show you what I mean on the next slide here in a second. All right. This is going to look complicated, but it's not. So you start in the middle and you work your way out. Because remember, each word is 3 letters long, and each word specifies a given amino acid. So if I ask you to transcribe, I'll write it down here. Make something up. I said, all right, what is GCU code for? Well, we start here. Okay, here's the G. And up here, okay, here's the C. And then, okay, here's a U. So GCU translates, that codes for alanine. Now, when you're studying, I need you to memorize each one. No, I'm just kidding. You don't have to do that. But on the exam, again, I will probably give you a strand of DNA. I will ask you to transcribe that strand of DNA, so then you'll have a strand of RNA. And then I'll give you something like this and say, all right, now I need you to translate it. And that's what you'll do. GCU codes for alanine. Does that make sense? So someone else give me, I think so. I just came up with a different one. I don't know. UUG. So let's see here. All right, there's the U, top right quadrant. Then you scoot up here. There's the other U. And here's the G, and that codes for leucine. That's how it works. And as I'm about to tell you on the next slide, you can see it in writing. This is all universal. So if you take a gene out of a human and put it into a wombat or an orange or a bacteria or a palm tree or whatever living thing there is on Earth, if you take a gene out of us and put it into one of those living things, it will code for the same thing. So in the human body, in cells in the human body, GCU says bring me alanine. UUG says bring me leucine. That's the same way for every single living thing on Earth that we know of. There might be exceptions, but if there are, we haven't found them yet. I'm interested one day, surely, with mutations and natural selection and evolution, one day there has to be some sort of change. But I don't know, it's been conserved for billions of years now, so that's just me thinking sci-fi stuff. Anyway, everything I just said, here it is in writing. Excuse me, like I said, the genetic code is universal. Actually, I did put a star next to that because there are some exceptions. You can look that up if you want. But what are the very, very, very rare exceptions for this genetic code being universal? Again, like I said, it's shared by the simplest bacteria to the most complex plants and animals and everything. In theory, with gene therapy, we could have your body producing whale proteins or mushroom proteins or scorpion protein, whatever the case may be. It's all universal. And basically that's what GMOs are. Well, some. For example, you don't need to know this, I'm just talking. People are so afraid of GMO, and I get some of it, but the rest of it I don't get. So there's a rice, I think it's called super rice. I forget the name of it. But it's just regular old rice. And as you know, in some parts of the world, that's mostly what they eat. They don't have much else to eat, right? So they eat a lot of rice. So what someone did was they took the vitamin A genes from carrots, because we need vitamin A, it's important, right? They took the genes from that and they inserted it into the rice, because rice doesn't make vitamin A. So now we have this whole strand of rice that also provides vitamin A. To me, that's not scary. Like, you're fine with eating it in carrots. What's scary about eating it in rice? Also, I've always had this idea, it's like my brother lives in Seattle, so marijuana is legal there. So I thought, how great would it be? You could probably make a ton of money if you could get the genes responsible for making THC and put them into other fruits and vegetables, right? So you can make like tomatoes that produce THC that'll get you high. Not that I want to get high, I'm just saying there's a market for it. Have like salsa that gets people high. Again, looking at it from a business standpoint, I'm not trying to do it. Anyway, that's all that stuff right there, right? It's fun. And if you want to, even for independent work, here's a paper you can write. It doesn't even require, well, no, I guess it does require a little bit of research. You can just think of your own crazy things that you would like to mix up. Like, what are some genes you would want to put from one species to the next? If you can think of it, go ahead and throw it in there. It'd be like capsaicin. That's the gene that makes things, or the capsaicin is the stuff that makes things spicy. But if you have like, I don't know, spicy chocolate or something, who knows? The world is your oyster. Write about it if you want. Anyway, another thing your book points out, this is important too, the universality of the genetic code suggests that it arose very early in evolution. We'll talk about that in later chapter, right? This is evidence of the fact that we all evolved from a common ancestor. But you have to believe that. There is evidence to support it. That's one of the evidence. Anyway, let's look at a picture. Here's some examples. Anybody, it's really hard to see with this, but one of these pigs is not like the other. So what do you think? Which pig is different, if you can see the difference? Is pig A, pig B, or pig C? Which one's different? You can tell. It's pig B. That's really hard to tell with this lighting. But this pig can glow in the dark. because they've taken an enzyme or a gene from jellyfish that glow in the dark. They inserted it into that pig, when it was just an embryo, so that mitosis made the perfect copy going on, and so forth. And now that pig can glow in the dark. Same thing with this mouse or rat, I forgot which. They inserted a gene from the jellyfish, and they can now glow in the dark. And it sounds like some crazy scientific, like mad scientist stuff, but it's actually for a purpose. It's hard to get into, but basically, if you're tracking a protein and want to see, doing experiments to see if this thing affects that protein production, then the way you can tell is if it glows or if it doesn't glow. But anyway, and then here's a picture that's not real, but people have suggested doing this, and it's just as feasible as this. You can take the gene from a protein that makes it glow and put it into trees, and then the trees will glow. And I guess the idea here is to eliminate the use of streetlights. But ecologically, there would be big problems with that too. But I don't want to get on that soapbox. Anyway, again, the point is the gene, the codes, the genetic code is universal. You can take genes from one species and put them into the next. So we'll finish 3 minutes early, but I want to start getting in because we're done with the overview. When we get back, we're actually going to jump into transcription. Like we're going to look at it in detail. So I guess the last word for attendance will be players. And again, same old thing. If you're watching this video later and you're not watching this live, then send me a sentence that uses the word players. If you're not a Cast fan, that should be easy. You can talk about Players Ball or all kinds of great songs. That's it. You guys have a great weekend.